Great. Thank you, Ajay, for that introduction. And it's great to hear that so many of you have registered. Thank you for joining us today. So as Ajay said, my name is Sucharita and I'm a curator for the Complex portal. And I also have Kalpana here, who is our Intact and Complex portal team lead. So I'll start with an introduction to the Complex portal, with starting with how we define a complex. The reason that this is important is that the definition of a complex can vary between databases. Then I'll take you through to the type of cross-references that you'll find for our complexes, and the type of structure and functional data that is associated with the complexes. And for those of you that are really familiar users with the Complex portal, you would have noticed some really dramatic changes to the portal over this last year. So new to the Complex portal, for the very first time, we have machine learning predicted human complexes. They were introduced late last summer. So we have now over 15,000 high confidence predicted complexes from the HUMAP3 and the music projects. And you would have also noticed that we now have an enhanced user interface. This is through a new tool called the Complex Navigator that was developed in-house, and it helps you compare the relationship between complexes. And finally, we have now some enriched annotation to the protein interaction data. So now we have little heat maps that provide information on protein-protein interaction, and this is sourced from Intact, our sister database, and also Proteome HD2, which I'll describe later. So what is the Complex portal? The Complex portal is an encyclopedic resource of macromolecular complexes. It can contain proteins as well as small molecules and other molecules such as nucleic acids, as long as they're a key part of the complex. And we now hold manually curated complexes across many model organisms. And importantly, as with all other EBI resources, all of this information is freely accessible to you. So all the complexes within the portal are tagged to many other identifiers from other resources. And looking at this, you'll recognize the icons for many of these databases. So for example, all of our protein components within the complexes, they tag by with UniProt identifiers. And the complexes are enriched with information from gene ontology for gene ontology component, function, and process terms. And this tells you information about the role a complex plays. And of course, we have physical interaction evidence that the complex really actually exists from interaction data from Intact. And then we have a reactor widget, which tells you about the biological pathways the complex is involved in. And the structural information comes from PDB and EMDB. And of course, all of the information, all of the literature information that we reference is tagged to EPMC. And we are also very grateful to our partner databases who contribute complexes on other model organisms. And these are Flybase, Wormbase, and SGD, which is a Saccharomyces database. So why do we need the Complex portal? As per the last slide, what's so great about the Complex portal is that it, by referencing from across lots of other databases, it helps you build up a comprehensive picture of a complex all in one place. So while we do have a great resource like UniProt, UniProt tends to focus on individual proteins and their function, whereas the Complex portal focuses on the complex as an entity. And just to mention that, like all other EBI resources, we have stable version identifiers for the complexes, so you can always keep track of your work. And I'll come to this later, but the manually curated complexes in the portal have also served as a really useful training set in within workflows which generate machine learning predicted complexes. So as I mentioned, we have manually curated complexes across several species. We now have it across 28, and we have the complete first draft of complexes for the Saccharomyces cerevisiae and E. coli K12 strains. So when we did our last release, we had about over 2,300 complexes, and now we have over 15,000 predicted complexes for humans. So we tend to release about 4 times a year. So another release is coming shortly, so keep a lookout for it. So the next slide is going to show you a little bit about the type of complex components you'll come across and the type of complexes you'll see in the portal. So I'd like to go over this definition of a complex again. So a complex is a stable set of 2 or more interacting molecules, and typically at least one of them has to be a protein, and the assembly should have been demonstrated to form a biologically functional unit. But recently we've extended this definition to include molecules which associate to form scaffolding structures where the same complex is ready to recruit other context-specific effector proteins to activate functionally distinct signaling pathways. So why did we extend this, and what does it all mean? It's simply because we found that there are lots of contexts that affect their that mediate their effects through molecules through different pathways in a very context-specific way. And I've got a really nice complex here to illustrate this. That's the mitosome core complex. So the mitosome becomes activated, becomes formed when the toll-like receptor pathway is activated, and it goes on to trigger a pro-inflammatory response. But when the mid-88 components that you see down here bind to IRAK4 to form the core, the size and assembly for this core complex is really dependent on the cellular state, and also whether it then recruits IRAK1 here or IRAK2 to mediate its effects. So, as I mentioned, other than proteins, you will also find small molecules in complexes, and they can be things like fat, NAD, or heme that you'll see in hemoglobin complexes. As well, complexes could contain DNA, RNA, or mRNA that you'll find in spliceosome complexes. So here are some examples of complexes and their components. So in the top left, we start with a GARP complex from C. elegans, and here all of the components are proteins. And then moving to the top right, we have the telosome complex, which is a Saccharomyces complex, and here you have the protein components interacting with telomeric DNA. And in the bottom left, we have a Cry1 mouse complex where the mouse proteins are interacting with zinc, which acts as a cofactor. And finally, we have another Saccharomyces complex, in this case a helicase complex, where you have 2 subcomplexes interacting to form the larger complex. So you have the MCM complex here, and you have a GINS complex here, each acting as a participant in a complex. So, as I mentioned, we have manually curated complexes across 28 species. So for the larger multicellular organisms, we capture them at the organism level. So you'll just see a Homo sapiens tag or a Mus musculus tags. But for the microorganisms, the complexes are captured to the model organism strain or reference proteome. So although we have focused on model organisms, we also capture non-model organisms if they're of scientific interest. So you would have seen several SARS-2 related complexes in the portal. And one other sort of unusual combination you would see is also some host pathogen mixed species complex. Here, this is because we can assign complexes to one species to make them more easily searchable. So when we have a host pathogen complex, the complexes are tagged to the pathogen species. So here is a example where you have a human ACE2 protein interacting with this SARS-2 complex. So all of the complexes within the portal are based on, have confidence scores assigned to them. And the confidence scores are based on evidence and conclusion ontology codes, where you'll see little links in the portal that'll take you to the ontology lookup service that'll tell you a little bit more about what these categories of evidence means. But basically, they help categorize the strength of evidence we have for a complex. And just to make these codes easier to translate, we use a 1 to 5 star rating system. So some of you older users would remember a system where we had these flask icons. I hope you find this a little bit easier to use. So all of the manually curated complexes in the portal can have between like a 5 and a 3 star rating. But the predicted complexes will have either a 1 or a 2 star rating. So what does it mean if a complex has a 5 star rating? It means that we have really strong physical interaction evidence, usually coming from a single experimental source. So for example, if it comes from the INTACT database, we'll have strong physical interaction evidence, like in the form of an SPR experiment or column purification experiment, or it can also be from structural databases like PDB and EMDB. And the complexes that have a 4 star rating, they're also based on experimental evidence. But for example, they would have been inferred from other related complexes in the case of paralogy, or across species in cases of orthology. And complexes where you see this 3 star rating, it means that the source for the proof for this complex comes from more than a single experimental source, and it's been built up by the curator, who's pieced together evidence from multiple weaker experimental sources. So when you search for complexes and you go into the details page for a complex, this is what you'll see. Here I've got an example of the 20S proteasome complex. So in the top left, we start with a recommended name. This is the name that a curator assigns based on how the complex is popularly referred to in scientific literature. Then you will also see a species name with a tax ID. Then you have a unique complex portal accession number assigned to it. Then moving along, you will see this table. It's a participant table which tells you all of the components that exist within a complex. So these are all protein, which are protein components. So they have this stable versioned UniProt identifier. But if the complex did contain small molecules, it would be referenced to EMBL-EBI identifiers. Or if it contained nucleic acids, it would be referenced to RNA central identifiers. Then we can look at the experimental evidence. So this complex has a 5 star rating. And as I mentioned, if you clicked if you clicked on this link for the evidence, it will take you to the ontology lookup service, and you can learn more about the categories of evidence. And the link underneath for INTACT will take you directly to the INTACT website, where you can pinpoint the experiment from which this evidence is based on. If we have information about the structure of the complex, then we're able to assign it stoichiometry. So you'll see numbers down this column. And if components of the components of the complex have specific biological role, for example, they're known to be catalytically active, like this PSMB5 and PSMB6 subunits here, then you'll see a little enzyme labeled tagged. Finally, we have the topology. So you can see that this complex is made up of 4 heptameric rings, and you can look at the binary interaction between each of these protein pairs. And if you clicked on an individual participant, it will tell you the binding region as well. So just following on from that last point, here is a much simpler complex to look at. We have the shelter-in complex with an expanded view showing you the binding region between the participants. So also what you would see on a complex's details page is a free text function statement. This describes what the complex actually does in physiology and where you might find it. It's tagged with gene ontology, molecular function, and process terms. And clicking on these links will take you to the Quick Go website where this is described in more detail. And if components of the complex have a enzymatic role, then you will see like RIA identifiers here or EC numbers, which if you click on this, it will take you to the XPC website. So other information you'll also find on the details page is free text on the properties for the complex. And by this, we mean the physical characteristics and the topology, etc. This extends to the complex assembly, which will tell you whether the complex is a dimer, tetramer, etc. Then you also see the reactome widget that I mentioned, which shows you the pathways the complexes can be involved in. And here's just a little quick tip I have for you. For example, if you want to search for complexes based on very specific function, process, or even Go component terms, you can search using a gene ontology number here. So for example, if you search with this function term, tumor necrosis factor production, then you should get a nicely tailored list of complexes that only take part in that function. So I think I've already mentioned the property statement and the type of physical characteristics of the complex that we capture. We also, if it's available, capture structural information. So here you can see all these PDB identifiers. And if you could scroll down, you could also see some EMDB identifiers if it's applicable. Then next to the identifier, you see this second column with a qualifier link. So what the qualifier tells you is that is whether the structures are an identical match or they're just part of a complex. And if you follow these links, they take you to the molecular interactions section within the ontology lookup service. And this describes this terminology in a bit more detail. And also on the complexes page, if it's applicable, you'll find a free text statement for diseases. And this will either be free text and or cross-references. And the cross-references will be to either experimental factor ontology, EFO, Mondo, or OMIM. And I should stress here that with complexes, we are very cautious in assigning them disease tags because we look at to see whether the complex as a whole is involved in a disease process, not just individual proteins. And if we know that a complex is being evaluated or looked at as a drug target, like many of the cytokines that you will see in the database, they will have a Chembl target ID as well. We also capture information on gene expression for the complexes components, and this is in the form of an expression atlas widget. You can just click on it and follow the links to that website to learn more. And under this widget, you will see the Go cellular component term. This will take you to the Quick Go component site where you can find out a little bit more about the complex term. For example, if it's tagged with a spliceosome complex, then it will tell you the type of components to expect. These all help. These all are ways to define the complex. And the other thing that you might see is a subcellular location for the complex. So this is one of the new features to the complex portal that I mentioned earlier. So we now have protein-protein pairwise interaction data. We've brought this in from both intact and Proteome HD2, which I'll come to later. So here is a really simple example again with shelterin, and I've clicked on the intact. You can just easily switch between these two. And in this heat map, the X and Y axis display the gene names. You have this correlation key underneath the heat map which goes from 0 to 1, where 1 is 100% correlation. And then if you hovered on any of these tiles, you would see a correlation coefficient for that particular interaction. And if you clicked on a particular tile, it will take you to the underlying data from either intact, if you're in the intact tab, or Proteome HD. This is really especially, I think, useful for complexes where we have that 3-star rating where you know the complex exists, but we pieced bits of evidence together, so you can look this up in Intax to see the type of experimental evidence that we have for those type of complexes. So the next few slides are going to focus on how best to search and use this resource. So I'll start with the home page where you see the search bar with quite a few examples of the type of search functions you can perform. So you can either search by UniProt ID, or you can actually feed it a list of UniProt identifiers. And you can also search by the gene ontology identifier, if you know it, or you can search by a complex name. But if you do that, I'd recommend that you put quotation marks around it to get a nice tailored hit list back. If you are interested in complexes from specific species, then you can just click on the organisms and directly select what you're interested in. And all of this information on the search possibilities you'll find in the documentations tab here. So if I did a search all with an asterisk, I get this whopping big list with 20,000 complexes. It looks a bit overwhelming, but you can filter this. So starting with the complex type filter, you can select whether you want to look at curated or predicted complexes. And then you can also slide this confidence score tab up and down, and according to the level of confidence that you're willing to accept for this complex, or you can use this species filter here. And so this is what we call the list view when you have a lot of complexes, but you can toggle between this list view and this navigator view. Just click on either of them. So this is the Complex Navigator. So this was developed in-house as part of an internship program by Susie Huget here. So this is the result of a search that I did for hemoglobin complex, and I've clearly put a human filter on it. So you see that the navigator is actually a matrix, and you have all of the row names here containing the complex names. And the complex components are contained within these columns. And you've got the complex components have little numbers inside circles, and what that is telling you is the stoichiometry for that component. So if these circles are left blank, that means we don't have structural evidence for that complex. But if you see a range, it means that the molecules for that component can vary. You also have this nice little basket feature that are going across the top here. So you can add individual complexes to the basket for just a closer comparison. And in this navigator view, you can fit about 20 complexes. If your search results returned a lot more, there will be a little next button to see the rest. So here are some more filtering options to help tailor your results. So you can use this sorting component box here. So by default, all the complexes that are similar in composition, they're clustered together so that the component order is optimized to compact clusters where the subunit is shared across complexes. Then you can also sort by organism. So you can have all of a particular organism bunched together, or you can sort by type, which groups whether they're protein or they have small molecules together. Then you can also change the display option. So you can either have a detailed display, like what we have up now, or you can have a simplified display. So if you want a simplified display, just deselect everything. And then if you add the component accession, then it adds back the UniProt identifier under the protein in this case. And if you click on the organism, you see this little human icon. And if you click on type, I'm not sure how visible it is, but this icon tells you it's a protein, and we have a little ring structure if it's a small molecule, and so on. So in the next few slides, I'll go over some nice examples where which demonstrate what you can do with this navigator tool. So it's great for comparing paralogous or similar complexes within a species. So normally proteins within a complex are captured at the canonical level, except if we think that there's a functional difference, we would capture them at the isoform or variant level. So here I've done a search for interleukin-6 complexes, and I've put a human filter on it, but it's returned with 4 different complexes. So why have we got 4 different complexes? It's because when interleukin-6, IL-6 here, binds to a membrane-bound form of its specific receptor alpha, this MIL6RA, and a membrane-bound form of its IL-6 signaling component, it forms a complex which sets off a classical signaling pathway leading to an anti-inflammatory reaction. And so it's tagged with specifically to demonstrate this with the Go function and process terms. And you can also see the pro-chain for this complex. But we have another example where interleukin-6 is bound to a soluble form of its receptor alpha and a soluble form of the signaling component. And it's formed a completely different complex, which has a totally different biological role. But this is a buffering complex, which stops trans signaling or pro-inflammatory pathways. And it also has a unique pro-chain tag, pro-chain assigned to it, and unique Go function and process terms. So the other use case for the navigator is to help compare orthologous complexes. So as I showed you before, we have manually curated complexes across 28 species, but we obviously don't have every single complex across the species. So in the Complex Navigator, we have this really nice tool, little switch called the Group by Orthologous. And here I've searched for human hemoglobin complexes. And for this particular component, hemoglobin D, with this toggle switched on, and if we expanded up the group members, you could see the orthologous proteins for to the human protein across other species, everything from chimpanzee to rat. And this information is brought in from the Panther database. The third use case I wanted to show you was looking at the relationship between complex and its subcomplex. So I should first mention that in the complex portal, we call something a subcomplex if the subcomplex has a biological role that is independent of the larger complex, so meaning that it's not just a fragment of the larger complex. So we have this adenosine methylase transferase complex here. And I've expanded this view, but you can see it here. Here is this subcomplex, which is expanded, and this smaller metal 3, metal 14 subcomplex is displayed here. And this relationship is also captured in the complex viewer, where we have the metal 3, metal 14 subcomplex with this identifier interacting with this larger complex. And again, this is reflected in the participant table, where you have this larger complex in this orangey color. And I've expanded this table to show the components, so you can see the smaller subcomplex in green here. So finally, the predicted complexes. So as I mentioned, we now have over 15,000 predicted complexes for humans from the HUMAP3 project and the MUSIC project. Hopefully, it's quite easy for you to distinguish between the two. The predicted complexes are in this lighter teal color, and you can filter for them using the complex type, as I mentioned before. So here is an example of a predicted complex from the HUMAP3 project. So you will have a complex identifier here, and also a link to the HUMAP3 project where you can find out a little bit more. So as I mentioned, all of the predicted complexes have a 1 or 2 star rating. At the moment, they've all been given a 1 star rating. And this is because they come from a single computational evidence source. But as we are moving more and more into looking at machine learning predicted complexes, if we were to bring in other sources of machine learning predicted complexes with evidence, we would then upgrade these ratings to a 2 star rating. So I'll just tell you a little bit about the HUMAP3 complexes. So the majority of the predicted complexes, over 15,000 of them, come from the HUMAP3 project. And this was a really nice collaborative project between us at the EBI and our 2 partner labs. So one of them was Kevin Drew's lab at the University of Chicago, and the other was Gail Kristash's lab at the University of Edinburgh. So to start with, we have these manually curated human complexes from the complex portal. So these served as a training set for generating this model. We then have physical interaction data, which is the HUMAP component. This is based on over 25,000 mass spec experiments. And Kevin's now published this paper, so you can read about it in this publication. And this covers 70% of the human proteome. So this was taken together with supporting evidence from the Proteome HD project, which looks at pairwise interaction data. So this is based on repurposed mass spec runs, and they look to find responses in expression pending on physical perturbations, such as a change in temperature, pH, addition of an agonist. Then this data was modeled with the HUMAP3, and the high confidence predicted complexes were then integrated back into the complex portal. So how is this integration done? So what we did was we took the predicted complexes and compared the components of the predicted complexes to the manually curated complexes, and anything that was an exact match was just merged in, but anything that was a partial match was added as a subset. Anything that didn't match was added as their newly predicted complexes. And then we're continuing to curate and validate these predicted complexes where possible. But the whole point of this project was an effort to map the human complexome. So we really hope to continue working on this and improving and refining this model, and also hopefully move on to other species. So the other predicted human complexes come from these multi-scale integrated cell maps. So this was a collaborative effort between Trey Eidecker and Emma Lundberg's labs. You can read more about it. This work's already been published. What this looks at is they've taken imaging data from the Human Protein Atlas in the form of confocal and immunofluorescent-based microscopy images. And they've integrated this with biophysical interaction data in the form of APMS, and they've converted this to generate a network that shows you a cellular hierarchy. So the music maps are also shown in this light teal color, and we have 317 music complexes, and all of the music complexes have an identifier that looks like this. And if you click on it, it takes you to an external Cytoscape application, which you can explore. And I should just quickly mention that all of the data from the music map is based on two very specific cell lines, the HEC293 and the U2 osteosarcoma cells. Just before I show you how to access complexes in the portal, I should mention that we're always really happy to take requests on complexes that you'd like to see appear in the complex portal. So please feel free to click on this request complex box. It's a really easy form to fill in. So to access the complexes, you click on this download button, and then it takes you to several options here. So the simplest option, which is a tab-limited file with one file per species, and that you can easily pass into an Excel file, is this complex tab format. So when you open it up in Excel, you'll see like a complex per row with about 19 columns, starting from the complex identifier down to the type of assembly for the complex. If you would like a richer annotation file for downstream analysis, you have this PSI MI XML 2.5 and 3, but the 3 is recommended. You can also interact with us programmatically, and there are 4 REST API methods here. There are also several points here that you can click on to learn a little bit more about your downloading options. Finally, I'd like to thank the Complex Portal team, past and present. It really is very much a team collaborative effort, and it's a lot of the content and how it was shaped is driven by Sandra Orchard here from the UniProt team, and Wan Ho, who is our developer for the portal and also did the hard work of integrating the predicted complexes, and Susie, who worked on the Complex Navigator. And thanks also to our partner databases who contribute complexes from other model organisms, and our funders here. And thank you all very much for listening. I hope it was an information overload. We are really easy to contact using the forms on our homepage, and we welcome community feedback. And you can also keep in touch with any news on training, and also follow us for our feeds on Complex of the Month. So we're now on Blue Sky, LinkedIn, and X. So thank you.
Great. Thank you very much, Siddhartha, for the webinar today. And thanks everyone for joining. So we got new question. Is there any way to explore the type of interactions between protein and other molecule interactions using this portal? So not just protein, but protein with something else.
You could search with a small molecule name. So, for example, do you mean like you want to search by like a heme as a cofactor or something like that? You could put inverted commas and search by the name.
Maybe I can also add, like when you do a generic query in the complex portal, you can also make use of the filter using small molecule as one of the component. And so then naturally it will bring all the complexes that has small molecule as one of the component. Yeah.
Yeah.
Great, thank you. Next one is, can you determine the interacting amino acids residues between complexes? Also, does the portal show drug interactions?
No, because we don't show drug interactions because we capture complexes in the natural physiological state. We're not a disease or drug sort of interaction. We're not that type of a database. With the amino acid level, you have your UniProt identifiers. I mean, I think you can when you look at the participants. Kalpana, were you going to?
Yes, I
Yeah,
can step
sure.
in.
You can step
So,
in. So.
yeah, okay. So, right now, the complex portal is using the experimental evidence on which amino acid domains are interacting with the neighboring partner. And we also planning to bring in all the IMEX annotations from intact, so from intact and for the IMEX curation that has been done for more than 2 decades. So this will bring all the binary evidences, particularly the binding domains between any 2 pair of complexes that can be loaded on to your complex and can see what's going on. So it's if you're asking me how to determine it, like we are planning to bring only the experimental evidence, but determination, if you are planning for some prediction model, you can use complex portal annotations as your training set.
And there's another one. What is the expected life cycle of the project? Will there be a version 2, etc., like UniProt? So I think it's.
So this is with the HUMAP project. It's been a really successful collaborative effort. I can definitely say that. And we are looking at funding options to continue the project. So in the short term, it will carry on till next year. But after that, we are exploring our funding options.
Oh, the complexes are here to stay. And what
Complexes will
we
stay.
do
Sorry, they are stable. Yeah, but
what we do further
the
is
next model is dependent, but yeah.
Okay, so someone is asking, is the paper released?
We do have a
Reference?
recent publication. We can share that with you later. We have a publication that came out earlier this year, a nucleic acid research describing the complex portal, the new changes to it, and the introduction of the predicted complexes. And we do have smaller little research projects going on within our team. So there will be other publications based on data from the portal.
Chichita, can you show in complex portal how to access all the list of publications? I think people would be that would be more useful.
So,
yeah, in the about us, and there's a publication stamp. Yes, and
this is the
Google.
latest one.
Yeah, you can click on the Google Scholar profile for the entire list, and yeah. This is for, yeah, so the latest, I think it is not updated yet, but I think
So
it'll be, yeah.
yeah, it'll be done here somewhere.
The latest publication is displayed on the website itself.
Yeah,
which
so
is in our
that's the
paper.
latest
Yeah.
one.
And the list of other further publications can be accessed in Google Scholar.
Great, that's good. Someone is interested in MD simulations. So I don't know if there's any application within the Complex Portal website where they can do MD simulations using the data.
We are yet to think about it, and thank you for the interest. I think it will be for the users to explore the applications, and what we are interested right now is providing the high quality complexes for anybody to explore to their prediction model or any simulation model. Yes.
Okay, so if there are no further questions, I would like to thank again Sucharita for the webinar today, and thanks Kalpana for the support, and thanks everyone for joining.